Okay, Max. So we've got our B cell model. It's working great, selecting B cells. We've got our progression markers defined, and we can see the various markers and their modulations. So now what I'd like to do is make sure that my model is as accurate as possible, really showing me where those transitions occur. And then I'd also like to be able to look at how a group of files relate to one another, get statistics on the group. Can we do that?
Yeah, and we just actually published a paper that uses a lot of those techniques.
Oh, cool.
Um, and just for everybody out there, the paper um, is in cytometry part B, and it's called Human B Cell and Progenitor Stages as Determined by Probability State Modeling of Multidimensional Cytometry Data. Can you say that 3 times really
No.
fast? No. And it's kind of embarrassing to me because it took me many years to realize the most accurate way of determining exactly where these modulation boundaries are. And it was such a simple solution, but it ended up being about 5 times more accurate than the way we had done it previously. And it's really that insight that led to that paper and studying what the real stages are in B cell ontogeny. So let me show you how this works. So we'll go ahead and launch GemStone again. And if you recall, Ben, we sort of left it in this state.
Right?
And what I'll do is I'll just go ahead and reanalyze this first file with the model that we had created. And so it selects on CD19, side scatter, and CD22. And now it's going through and stratifying the B cells on the basis of CD10 downregulation, CD34 downregulation, and then the 3 levels in CD45. So this is what we ended up with. And now we want to actually make some tweaks on this so that it's actually as accurate as possible. When I'm saying that, what I mean is that we can find exactly where this CD38 downregulates in relationship to 10 and 45 and so on and so forth. The technique is quite simple, and simpler is better in this case. So let's just take a look at CD10 for a moment. And if I take this control definition point, or CDP, and move it around, you can see that as I move this away from the next CDP, it goes a certain distance and then no more, the distance between these.
Mm-hmm.
And that's what we use to model the transition from one state to another. Now, what we want to do is we want to actually make it so that there is no transition between them. And by doing that, we'll then model, it's kind of like the midpoint of where the transition is, and that ends up to be really accurate. And then by making it accurate, we then can ask some really nice statistical questions about whether 2 modulations are at the same point or not.
Okay.
We can address that statistically. So the way we do that is we go into the quick editor, and we just, where it says maximum transition, we just set it to 0. And as soon as I do that, you can see these snap together. And now if I move this around, it's
Okay, so
all
the
locked
X
together.
distance between them is 0.
Exactly.
Yeah, okay.
So I could go through the entire model and do each one like this, or I can just come up to preferences and go down to parameters, and right here I can just make sure that that's set to 0. So I'll just force it to change here, and then we'll say OK. So let's just go through and check to make sure it did what we want. So here's CD34, and you can see that's locked so that there's no transition. And then the same way with 45, 3 levels.
Yep. Okay.
Okay, so that's the way we can make it as accurate as possible, which was part 1 of your question.
Yeah.
Part 2 of the question is, okay, we want to analyze all these, but we want to gather statistics on all the files that we're going to analyze. How do we do that? And so we'll do that at the same time here. We're going to go to Analysis, and we say, go ahead and initialize and start stats. So that becomes part of the model. So when that model is loaded in, it's going to clear out the statistics and then start gathering statistics as we analyze files.
So as long as that model is loaded, it will sort of aggregate the statistics of files that it sees.
Exactly.
Okay.
We've got a few other things to do to make this work, but at this point we'll go ahead and save that model. So we'll click on File, and normally what I do, Ben, is I hit Reset before saving, which puts everything back into its the same state it was in when we loaded the template.
Right.
And then I'll go ahead and save template, and like we have been doing all along, we want to increment the version and save it.
Yep.
So at this point, template's ready to go. Now we just need to do a little work here in the batch system. So when we hit level 2, we want to now load that specific template. So we'll change this 3 to a 4. And so that's done.
Okay.
But now when it gets to the next file, you see that's also on level 2. So if I leave it this way, it's going to reload that template.
And clear out the statistics.
And clear out the statistics, which is not what we want. We want it to accumulate as we go. So this is where we're really leveraging the levels, so to speak. So we'll go ahead and highlight these two, indent, and now in level 3, what we want to do is just do a reset on the model. So everything goes back to where it was when the model was first loaded, but the model stays resident, so it's creating statistics as it goes.
Okay. Sounds like a plan.
All right. So let's let it go. So we're going to go ahead and double click on the first file. And so again, it goes through the selection process, goes through the stratification, but now you can see that it's exactly finding that transition point.
Also seems to be moving through this a little bit quicker.
Yeah, because we actually simplified the model, so there's less for it to do. And that's simpler sometimes is indeed better. So it's all done.
Okay.
Fast. But now we go, all right, so let's go ahead and analyze the next file. It does the reset now because it's on level 3, and now it's finding all the exact transitions for this file.
So I could press one of the buttons on the toolbar at the bottom and just walk away from this, right? I can let it, we don't have to sit here and watch this do this.
We don't. So in the paper we had 16 normal bone marrow samples, and the only trick to that, Ben, is that at the end, so at this point right here, we would put a stop auto advance, and then we could just automatically analyze them
Okay.
all.
Because the files that we have after this are a different cell type.
Yeah, they're T cells, so we wanted to actually stop the advance. So we'll do the third one. And so at this point, it's just about done. And what I just want to show you, Ben, is where all the statistics is kept.
Right.
Because it's not intuitively obvious where it is. But we'll go into 10 here. We'll go into the same CDP that we were in before. We'll double click on it. We're interested in the horizontal locations in this case. So we'll click on state, and then we click on this button down here. And right here you can see, here are the statistics. It's showing that it has 3 statistics at this point. The lowest for this particular downregulation of CD10 was 50.77. Highest is 70.52. But here are the important ones. This is the mean, 61.5, and then this is the standard deviation, just about 10. The neat thing is that not only is it keeping track of mean and standard deviation, but it actually then, as we were going through the system, it was finding the correlation of this CDP to every single other CDP in the model.
Okay.
And keeping track of those statistics. And we can see that right here when we click this. And let me move this over so you can see it. So here are the correlation values between the CDPs.
Okay.
So if 2 CDPs are marking an end of a stage, they'll be highly correlated. So we're looking for a high correlation, and the p-value will be less than 0.01 or something like that. And then, so that's what all these do. Obviously, we only have 3 here, so significance is not going to be here until we get more in our sample. And then the other thing is that it finds the difference between the 2 CDPs and then subjects that to a paired t-test. So we can do both statistics at the same time to really answer the question whether 2 things are happening at the same time or not, and thereby create statistically determined stages. So that's the gathering statistics part of all this. And then the last part is, for the longest time, we always had to pick a representative file for our papers and stuff like that.
Right.
Now what we can do is we can actually show an average model that represents all the files that are in a specific group.
Oh, that's fantastic. Right.
And it's really, at this point, you can just imagine what it's going to do. It's going to take those means and standard deviations of each of the CDPs, and it's going to reset all the CDPs in the system to that mean value when we tell it to.
Right.
So in order to do that, we click on Analysis, and then right here, Ben, where it says Update Model from Stats, when I click that, you'll see the CDPs actually move into their mean position.
I see. So that is the mean of the set of 3 files that we analyzed.
That's right.
Okay.
And then if we want to look at the overlay for these means, now it's only going to show us the means of those things that were modeled.
Yep.
So in this case, we only modeled CD10, 34, and 45.
Right.
Under normal circumstances, if you were interested in really understanding the coordination of all markers, we would model all of them. But this just will give you an idea. We come in, at this point, this is actually showing us real data, the actual observed data. We don't want to actually look at that. We want to look at the model. And when I do that, we can just see the coordinated changes for the average model,
I see.
and then study, the staging for that.
Well, that's fantastic.
So that's it in a nutshell.
There you go.
Okay.